I hope you all enjoyed the DNA and other stuff game. I know Hunter probably did enjoy it quite a lot. You're joining me from Hilo, Hawaii today, and of course in Hawaii one of the best things is the local fruit like this papaya. So I decided we're going to extract some DNA from fruit today, and then we're going to use the papaya enzymes to help us to remove and degrade the proteins that surround and package the DNA. So remember that DNA is stored as chromatin, which is a combination of both DNA and proteins that package it. So let's go inside where I have things all prepped, like cooking show style, and we're going to extract some DNA. So what I did earlier was that I took our blender and just kind of blended up some really great local bananas. And I took those bananas, I put them into a bag, and now you can see we've got mushed fruit. It's really sexy. And at this point, what I'm going to do is I have half a cup of water that I got from the pure water filter over there. And then I'm going to add to this 2 tablespoons of dish soap. I don't have any Dawn, but this is Ajax. So it just happened to be here where we're staying. So 2 tablespoons of dish soap. I've added that into this mixture. And then I'm going to add in a teaspoon of salt. You're probably thinking about this right now, and it's funny how this brings together almost everything we've talked about this semester. Of course, dish detergent is going to emulsify the lipids. It's going to, dissolves with the lipids in the membranes of the banana. That's going to free the DNA. And now the salt's going to come in here pretty soon as well. But I want you to think about the kind of interactions that a sodium cation could be having with the anions of the sugar phosphate backbone, right? The phosphate anions of the DNA. Now we've got this solution all ready to go, and we're going to put some of it in with our banana. We may not need all of it, but we've got plenty to go around. So we're just going to put that in. And you can picture what's going to be happening as the cells in the banana begin to be dissolved by the detergent and free the internal contents of the cell. And of course, we're interested in the DNA. So releasing the DNA from the nucleus of the cell. And of course, remember, plant cells are eukaryotic cells, so they have a nucleus. We're going to free that DNA. Now, of course, you'll recognize that there's a lot of salvation spheres around the phosphate anions. Remember that of the DNA backbone. And so they're all salvated with that hydration sphere. And in a moment, we're going to talk about how we can bust up that hydration sphere so that the sugar phosphate backbones can interact properly with that sodium cation. Let's leave this for about 20 minutes. It's been 20 minutes for me, probably not for you, but we're ready to now filter our banana. So we're going to assume that the cells have lysed and the DNA has been freed, and we're going to put this through just a simple coffee filter. Now, of course, this is going to take some time to pass through, so we're going to give it just a little bit of coaxing so that we can get the banana juice. Great, so we finally got the banana juice all filtered. It took just a few minutes. Good thing for time lapse, right? Cooking show style. So now I'm going to add to this the papaya. This is juice that I took from the papaya just a little bit earlier. And our goal here, now you could do this with a pineapple too, to get the enzymes that we know are proteases that will degrade the proteins around which the DNA is packaged. Now remember when we talked earlier about papain being a protease within the papaya? So I thought it would be cool to use the papaya. So we're going to let this sit for just a few minutes, and we're going to allow the proteases to go to work on the proteins to free the DNA so we get a pure DNA extraction. Now we've hit the moment of truth, and we get to see whether this actually works. In the freezer, I've been chilling down some isopropyl alcohol that just happened to be in the bathroom. And our isopropyl alcohol will serve to extract the DNA. So what we need to do is to layer this on top, knowing that the alcohol with less density will lie on top of the water. And at the interfaces, we should be able to start to see the DNA extraction occurring. Think as I do that, think about why it is that the alcohol, a less polar molecule than the water, enables the DNA to stop interacting, right? Those sugar phosphate backbones. And you want about equal volume to the amount that we had of the initial liquid. So now this frees the sugar phosphate backbone to begin to interact with the sodium cations, and we get a precipitation. So the DNA should precipitate out into the alcohol. So alcohol extraction is a very, very common means by which we extract DNA even in the laboratory, not just in the kitchen. So let's see if there's anything that we start to see. Yeah, you can start to see some bubbles there, but around in those interfaces there's some white stuff forming, and this is where we're getting the DNA. And I can see if I can put this in here. We start to see the DNA. It's kind of this sticky layer that's coming off at the interfaces. And if we had a microscope, I didn't bring one with me to Hawaii. Oh yeah, there's some right there. We would be able to see it better. Maybe I'll turn my skewer. Oh yeah, there it is. So chromatin, the DNA, and the proteins that package the DNA. And this is so very important. It's hard to overemphasize how important packaging is. In fact, here's a good way to state it. If you were to take the longest known human chromosome, and you were to look at that chromosome in its bDNA form. So remember from the game that bDNA is this quintessential DNA molecule, right, that we see there. And you were to elongate that bDNA molecule, you would find that it would reach the length of 8 centimeters. So look at this. This is ridiculous. I've got a ruler right here with me. And if we look at 8 centimeters, we're talking a length from here all the way to here. That's how long that single longest chromosome, human chromosome, would be if you elongated it out, still leaving it in its bDNA form. Holy moly! And yet, upon condensing as a chromosome, we see that it can condense down to only 10 micrometers. So that is so tiny, right, comparatively to what it could stretch out to be in its elongated form. So packaging, what this tells us is that packaging is absolutely one of the most important things about managing DNA, whether in a dividing or in a non-dividing cell. In a non-dividing cell, chromatone is just simply going to exist as what often looks like long, thin threads with a diameter of about 30 nanometers, and we'll see in a moment where we come to that level of packaging. But humans have, as you guys already hopefully know, 46 such fibers, that is 46 chromosomes, sometimes known as 23 pairs of chromosomes, and during metaphase, as we just mentioned, these condense an immense amount. In fact, starting from their linear form to their fully condensed form, as we mentioned, a total of 8,000-fold condensation. Wowzers, how does it happen? Well, the first level of packaging is the formation of something called a nucleosome. And nucleosomes are basically the DNA wrapped around these small basic proteins called histones. Histones are very rich in lysine and arginine, which you guys know right off the bat are the basic or positively charged amino acids. So this overall takes on a positive charge, this histone packet. And what this is cool for, or I guess if you think about the interaction with DNA, remember that DNA is negatively charged. It is acidic, right? And so it will tend to associate and love to associate with these small basic histone proteins. Now, what happens with the histone proteins is that there are actually quite a number of different histones, H1, H2A, H2B, H3, and H4. And within the smallest sort of packaging unit, what we see is the formation of what's called a histone octamer. And in fact, the octamer is made up of 2 each of H2A, H2B, H3, and H4.
Just when you were thinking it must be time for a cheesy demo, you're going to get your wish, because a few years back I was back in my backyard and I was checking out the garden hose and I was like, man, that would make super good histone octamers. So my garden hose bit the dust and we ended up with a cheesy model that shows the packaging of DNA at its most core level. So what you see here is that the garden hose is meant to represent the histone octamer. Of course, it would be comprised of 2 copies each of H2A, H2B, H3, and H4. Wrapped around that very basic core is the DNA, and I'm showing the DNA in the neon kind of green color. And what you can see there is that it's wrapped 1 3/4 time around that histone octamer. Now, we recognize the DNA with its negative charge loving to interact with the basic amino acids, the positively charged amino acids in the core. So this actually constitutes the core particle. So what we're recognizing here is that we have the 146 base pairs of DNA wrapped around the histone octamer, and together we would term those the core particle. Now, that's not all there is to the packaging. We can see that holding together each one of the core particles is a section of DNA that is not associated with the histone octamer. And perhaps if we come down here, we can see that unimpeded. We've got this region here of DNA that we actually term the linker DNA. So the linker DNA is 54 base pairs long. And what you noticed up above here is that linker DNA is associated with yet another histone. This histone is called H1. So H1 associates with the linker DNA, and now we can define another term, that is, we have the very lowest level, if you will, of full packaging unit that repeats. We've got 200 base pairs now of DNA, including the 146 base pairs wrapped around the histone octamer, and then we have the 54 base pairs of linker DNA associated with H1. So all of that all together is considered to be the nucleosome. So the nucleosome is that region that is comprised of H1, the linker DNA, the 54 base pairs, the 146 base pairs around the histone octamer, and then the components of the histone octamer. And together, that nucleosome is what we're able to define there. So 1 molecule of H1, 2 molecules each of H2A, 2B, H3 and H4, and approximately 200 base pairs of DNA. Now, these H1 histones play a further role in packaging. That is because they tend to interact with one another. So in their interacting with each other, it causes further condensation. And we'll see momentarily what that will be called once all of the H1s begin to associate with one another and further condense this molecule. So we get a pretty sweet core level of packaging, and I'm glad my garden hose bit the dust. Really, it was a shitty garden hose anyway.
So the question down at the bottom here, what is the point in all of this? Well, we certainly know what the point is, right? It's all about condensation. And in fact, this achieves about a 10% reduction in length. So we'll just jot that down. And our first level of condensation is done, but it's only 10-fold reduction in length. So we're looking for something like an 8,000-fold reduction in length. So we have a little bit of ways to go before we see that we have gotten to the full point of condensation. Now, of course, remember, as I mentioned on the model, looking at the model, we do see part of that condensation having to do with the interactions between neighboring H1 molecules. Let's look at that now. And of course, the picture says 1,000 words. So here we see at the top of this figure from your book the different kinds of histones, H2A, 2B, 3, and 4, coming together to form the histone octamer. And then, of course, when the DNA wraps around that, we see that being termed the core particle. So the histone octamer plus the 146 base pairs wrapped around that octamer. And then associated with the linker DNA, we see the H1 histone, and forming the nucleosome is the linker DNA, the H1 histone, and the core particle together, and that is our sort of core packaging unit, the nucleosome. Now let's look at how these H1s will interact with one another and form a further bit of condensation. Not a lot, but enough. So neighboring H1 molecules will bind to each other, bring the nucleosomes together, and that's going to form something that is termed the solenoid, or the 30 nanometer fiber. So these H1s allow for a little more condensation by just simply interacting with one another, about a 4 times reduction in length that we can see there. So the 30 nanometer fibers help with the formation of those, help with condensation, but they don't do the entire trick. And from here, these 30 nanometer fibers actually form loops and attach to an RNA scaffold, an RNA protein scaffold. And this is actually one of the major forms of condensation. In fact, allows for 200 times condensation. And I can't help, whenever I think of this, I always think of coiling a rope, a climbing rope, because what you're seeing there is definitely a condensation of what was a very long rope, and you kind of coil it into a much more condensed form. And in some ways that's a lot what this is like, but attaching it to an RNA protein scaffold as well. So further, these loops that are attached onto the scaffold can condense by supercoiling as well, to allow for that full 8,000-fold condensation by the end of all of these different kinds of packaging. Let's take a look at a picture that really helps to visualize all of this. Here is the 30 nanometer chromatin fiber. You can recognize the condensation afforded by the interactions of the neighboring H1 molecules. Here we can further see the condensation in terms of attachment to the RNA protein scaffold, and if you look really close on these figures, maybe you dim the lights or something wherever you are, and you can actually see that these are loops that then attach back on to that scaffold, and it's a pretty cool method of condensation.
Organisms within the bacterial and the archaeal world also use proteins to package their DNA. And for those of you who remember studying microbiology or like to dork out with a little micro, you might recall that some archaea quite literally have histones. That is, for example, if you think of thermoplasma, which is one of our eukaryotes, it does package its DNA in the same sort of way that a eukaryotic cell does, using histone proteins. So the difference between the so-called prokaryotic world and the bacterial world is not really maybe as grand as we might have thought. But we can say that bacterial DNA packaging does tend to be a little bit different than that of eukaryotes, and bacteria do tend to lack histones. So whereas we do sometimes see those in certain archaea, we don't tend to see them within bacteria. They do have packaging proteins though, and certainly we're closing those dramatic gaps between the prokaryotic and eukaryotic world by seeing levels and shades of gray there, but certainly don't tend to be thought of as histones. So within the bacterial world, we tend to see the lack of nucleosome-like particles, but we do see extensive packaging, and certainly we do see the attachment of large loops of DNA to a scaffolding backbone. And this sort of condensation is fairly extensive also within bacteria. So this condensation, in addition to extensive supercoiling, converts the bacterial chromosome into the so-called nucleoid. This is my prokaryotic cell, and it's kind of that quintessential model of the prokaryote, where you can see the gel-like mass of what's called the nucleoid, where we see that condensed DNA and protein material that allows for a sort of a more succinct or discrete region where we see the so-called nucleoid.